Hello everyone, a very warm welcome to my botany classes. Today I'm back once again with a fresh new topic. Today we will be talking about Rhizopus. This is a very famous, very well-known fungus. Now before we start talking about Rhizopus, let's know about its systematic position. Okay, Rhizopus belongs to the division. See, before you study any fungal species, you must know its systematic position. You must be aware of that. Division Eumycota. Eumycota, subdivision Zygomycotina, then class Zygomycetes, order Mucorales, family Mucoraceae, here we will talk about the genus Rhizopus, and species Stolonifer. Okay, Rhizopus Stolonifer. Rhizopus Stolonifer. Okay, so the genus Rhizopus is quite large. You know, it has multiple species to it. Of these, Rhizopus stolonifer is very common and occurs frequently on bread and is therefore also known as bread mold fungi. Okay, this is also known as bread mold fungi. Know the name, please know the name. It is also known as bread mold fungi. Okay. If we talk about its vegetative body, the vegetative body is well-developed mycelium composed of long, slender, and much branched hyphae. See, if you take a look at its vegetative body, you will be able to see that it is composed of long, slender, much branched hyphae. The hyphae are cenocytic, that is asepted. So vegetative body, if we see, it is well-developed mycelium. I'm just writing the points. Well-developed mycelium composed of long, slender, and much branched hyphae. Long, slender, and much branched hyphae. All right. These hyphae are asepted. Okay, the hyphae are cenocytic. See, remember these hyphae are cenocytic. That means what? Wall formation hasn't taken place. Plenty of nuclei are there. Okay, plenty of nuclei are there. That means septa formation hasn't taken place. The mycelium of Rhizopus, very soon enters the reproductive phase and becomes differentiated into rhizoid, stolon, and sporangiophores. See, if you take a closer look at this, it soon enters the reproductive phase and gets differentiated into rhizoids, stolon, and sporangiophores. These are sporangiophores. Sporangiophores. Look here. These are the sporangiophores. Could you see this? These are the sporangiophores. These structures. All right. Stolons are subaerial hyphae that grow horizontally above the substratum. See, if this is the substratum, all right, if this is the substratum, the stolon is growing horizontally above the substratum. Rhizoids arise in tufts from each node. This is the nodal region. See, these are the nodal region. And rhizoids are arising in tufts, in bunches at the nodes. And it grows towards the lower side and play important role in anchorage. It helps the fungus to remain anchored on the substratum. Okay. Sporangiophores are erect, aerial, and unbranched hyphae which grow upward in cluster at the point where stolon forms rhizoids. So see, these are the points where stolon is forming rhizoids. From here, slender erect structures grow. This one, slender erect structure grows upwards, aerial structures which bear the sporangium at its tip. Okay. So this is the brief discussion on the vegetative structure of this fungus Rhizopus. All right. See, the hyphal cell wall is composed of microfibrils of chitin. Chitosan also may be present. Okay, other polysaccharides like glucosamine and galactose, proteins, lipids, etc. are also present. Fine. Hyphal cell within possesses granular streaming cytoplasm, vacuoles, and mitochondria of various sizes, numerous nuclei, endoplasmic reticulum, ribosomes, droplets of oil, and glycogen as reserve food. So what is the reserve food? Reserve food is glycogen. Cell wall is basically composed of microfibrils of chitin. Microfibrils of chitin. All right. This is what it is composed of. Fine. Now we will talk about the reproduction in Rhizopus. Okay, let's start with asexual reproduction. Asexual reproduction takes place by non-motile sporangiospores. All right. These are formed within the black-colored globose sporangia. Okay, these sporangiospores, the sporangiospores are formed within the black globose sporangium. All right. At the time of reproduction, one or more aerial hyphae are produced in cluster. See, at the time of reproduction, what's happening? One or more Aerial hyphae are produced in cluster directly above the rhizoids. These structures are known as sporangiophores, which we were just discussing. These structures, all right. See, it can be one or more. Here you can see one. Here you can see more than one. 1, 2, 3. Here you can see 2. And see, this globose structure is the sporangium. And it bursts open to give rise to spores. See these dotted thing, take a minute look. These portions, see, you will understand better. Okay. These structures are known as sporangiophores. The tip of the sporangiophore form globosporangium. All right. The central portion of the developing sporangia become highly vacuolated. The vacuolated zone gets separated by wall formation and the central portion forms the columella. Look, this is the central portion which forms the columella. Okay, see, this is the young sporangium. This is the vacuolated area which forms the columella. All right, the vacuolated region, they first get separated by wall formation and the central part forms the columella. Understood? The multicellular segments of the peripheral zone gradually secretes wall around themselves slowly and metamorphose into sporangiospores. This region was initially sinusoidal. That means nuclei were all free. See these structures, you can see, isn't it? See this, they're all free nuclei. No walls. Later, wall formation take place and they become, or they rather get metamorphosed into sporangiospores, right? Now, when they become matured, the spores get dispersed or discharged. The dried sporangial wall breaks up into fragments to liberate the dry spores, which are usually dispersed by wind. Look at this picture. Okay. Under favorable conditions, it germinates by germ tube, which develops into fluffy, much branched aerial mycelium. So these are the spores. See, they getting favorable condition, they give out a germ tube. Understood? So this is how we come to an end of the asexual reproduction in Rhizopus stolonifer. Okay, now we will look into sexual reproduction in Rhizopus. In sexual reproduction, see first what happens. Sexual reproduction takes place. Please don't write like this in your exam. This is for the purpose of explaining you. Sexual reproduction takes place by gametangial copulation. Remember this word, gametangial copulation method. Okay, what happens here? See, Rhizopus stolonifer. In Rhizopus stolonifer, you will see two types of hyphae of opposite strain. One is plus, the other is minus. That means what? It is heterothallic. Hetero means what? Different. Homo is similar. Of course, it is hetero, isn't it? Because two different strains are observed here. They come in contact with one another. Okay. Then what happens? After they come in contact with one another, copulating branches called progametangia are formed. See, the plus strain and the minus strained hyphae, they come in contact with one another. They come closer to one another. This is the minus and this is the plus. Thereafter, what happens? Progametangia formed by copulating branches. Okay, see, progametangia are formed by, copulating branches here. Next, what is happening? As the progametangia begin to enlarge, huge amount of cytoplasm and nuclei flow to the contacting tip. This is the contacting tip, right? This is the point of contact. See here. This is the contacting tip, this part. All right. So huge amount of cytoplasm flow to the contacting tip of progametangia, and the progametangia begin to enlarge. Fine. It is separated into two cells by a septum. The terminal cell forms the gametangium and the basal cell forms the suspensor. See, wall formation takes place. Forming gametangia, the terminal ones are the gametangia and the basal ones form the suspensor. Okay. The gametangia formed from the terminal cells and suspensor formed from the basal cell. I hope it's clear. See. Next, each gametangium contains multinucleate protoplast because wall formation hasn't taken place, which constitute an isogamete. Fine. It constitutes an isogamete. Iso means similar. Isogamete. All right. So what did I say? I'm repeating once again. Each gametangium contains dense and multinucleate protoplast, and such protoplasts constitute an isogamete, which is also known as xenogamete. Fine. This isogamete is also known as xenogamete. Okay. In the meantime, the walls of the two contacting gametangia dissolve at the point of contact, and the protoplast of the two xenogametes unite to form a new cell. Okay, now the contacting wall is getting dissolved, and once the wall gets dissolved, the protoplast content carrying plenty of nuclei, they merge. Okay. The protoplast of the two xenogametes unite to form the new cell wall. The new cell wall thus formed enlarges to give rise to thick-walled zygospore. This is the zygospore. Its structure is a bit warty. Okay. So zygospore has got a thick-walled warty structure. What is that? Yes, what is it? Thick-walled warty structure. So let's write so that you don't forget. Thick-walled warty structure. Got it? Next, see. Many zygospore nuclei fuse in pair to form 2N nuclei, which undergo meiosis just before germination. If it undergoes meiosis, what will happen? 2N will produce N nuclei. Diploid will become haploid, since meiosis is reduction division. Fine. Half the haploid nuclei produced by meiosis are plus-strains or plus mating type, while the other half is of minus mating type or minus strains. All right. As the zygospore germinates, see, as the zygospore germinates, the outer wall or exosporium cracks open, and the inner delicate wall or endosporium protrudes in the form of a tube called promycelium. Clear? That bears the germ sporangium at its tip. Fine. The germ sporangium constitutes the sporangiospores, either all plus strains or all minus strain or both types. The spores, after liberation, germinates into a fresh germ tube which produces new mycelium. All right. So this is all about the sexual reproduction in Rhizopus. A different type of a phenomenon called parthenogenesis is also observed here. What is that? What is parthenogenesis? Let's talk about that a little. Let's know what is parthenogenesis. What is that? Gametangia, you know what happens, often fail to copulate, and such a single gametangium may develop directly into a thick-walled structure called parthenospores or azygospore. Let's write what is it? Gametangia. Gametangia often fail to copulate. Gametangia often fail to copulate, and as such, a single gametangium single gametangium may develop directly into a thick-walled structure, directly into a thick-walled structure called parthenospore or azygospore. What is it called? Parthenospore. Parthenospore or azygospore. Okay. This results when copulation doesn't take place. The gametangia fail to copulate. Understood? Parthenospore resemble the typical zygospore only. There is no much difference. So the external structure is similar. And that also germinate into a new mycelium in due course of time. So we can write that also, that germinate into new mycelium in due course of time. All right. Understood? Okay, now we'll just talk a little about its economic importance. We must always know what are the economic importances of this, of a particular genus that we study. Okay. Rhizopus stolonifer is used commercially for the manufacture of fumaric acid. Fine. And for some steps in the manufacture of cortisone. Let's just quickly write its economic importances. We must know. Economic importances. What is that? Used in manufacture of fumaric acid. All right. Fumaric acid. Next, what other? Alcohol manufacture. Okay, alcohol is obtained from Rhizopus or Rhizae. alcohol obtained from Rhizopus oryzae, Rhizopus oryzae. Okay. Other than this, various species of Rhizopus like Rhizopus sinensis, Rhizopus nodosus, they are all capable of forming large quantities of lactic acid, right? In Indonesia, a delicious food known as tempe is prepared from soya beans with the help of some strains of Rhizopus. The name of this Indonesian delicacy is tempe, T-E-M-P-E-H, tempe, Indonesian delicacy, Indonesian delicacy. Fine. Now, see, Rhizopus stolonifer causes rot of sweet potato or fruit of apple, strawberry, and tomato. So it causes a lot of economic loss also. This fungus is also responsible for leak disease of strawberries in storage. Okay. Rhizopus species cause fungal disease, mucormycosis of domestic animals and man. Fine. So these are some of the very important economic importances of Rhizopus. I hope you have understood the topic. If you have any confusion or any doubt anywhere, please feel free to contact me or put your doubts in the comment section. I will try my level best to clear them. Okay. Thank you so much for your kind attention. Stay connected. We'll be soon back with a fresh new topic. Till then, goodbye.